Chapter 3, Plant Kingdom. In the previous chapter, we looked at the broad classification of living organisms under the system proposed by Whitaker 1969, wherein he suggested the five kingdom classification with Monera, Protista, Fungi, Animalia, and Plantae. In this chapter, we will deal in detail with further classification within kingdom Plantae, popularly known as the plant kingdom. We must stress here that our understanding of the plant kingdom has changed over time. Fungi and members of Monera and Protista, having cell wall, have now been excluded from Plantae through earlier classification place them in the same kingdom. So the cyanobacteria that are also referred to as blue-green algae and are not algae anymore. In this chapter, we will describe algae, bryophyte, pteridophyte, gymnosperm, and angiosperm under Plantae. Let us look at the classification within angiosperm to understand some of the concerns that influence the classification system. The earlier system of classification used only gross, superficial morphological characters such as habit, color, name, and space, shape of leaf, etc. They were based mainly on vegetative characters or on the androecium structure system given by Linnaeus. Such systems were artificial. They separated the closely related species since they were based on a few characteristics. Also, the artificial system gave equal weightage to vegetative and sexual character. This is not acceptable since we know that often the vegetative character are more easily affected by environment as against the natural classification system developed, which were based on natural affinity among the organism and considered not only the external feature but also internal feature like ultra structure, anatomy, embryology, and phytochemistry. Such a classification for flowering plant was given by George Bentham and George Dalton Hooker. At present, phylogenetic classification system based on evolutionary relationship between the various organisms are acceptable. This assume that organisms belong to the same taxa, have a common ancestor. We know use information for many other sources to help resolve difficulties in classification. These become more important when there is no supporting fossil evidence. Now, numerical taxonomy, which is now easily carried out using computer, is based on all observable characters. Number and codes are assigned to all the character and the data are thin processed. In this way, each character is given equal importance. At the same time, hundred of character can be considered. Cytotaxonomy, that is based on cytological information like chromosome number, structure, behavior, and chemotaxonomy, that the chemical constituent of the plant to resolve confusion, are also biotaxonomy. This day, algae. Algae are chlorophyll-bearing simple thalloid, autonomic, and largely aquatic, both freshwater and marine organisms. They occur in a variety of other habitats, moist stones, soils, and woods. Some of them also occur in association with fungi, lichen, and animalia. Animals, example on sloth bear. The form and size of algae is highly variable, ranging from colonial forms like volvox and the filamentous form like Eulothrix and Spirogara. Figure 3.1. A few of the marine forms such as scalpus from massive plant bodies. The algae reproduce by vegetative, asexual, and sexual method. Vegetative reproduction is by fragmentation. Each fragment develop into a thallus. Asexual reproduction is by the production of different type of spore. The most common being the zoospore. They are flagellate, motile, and on germination give rise to new plant. Sexual reproduction take place through fusion of two gametes. This gamete can be flagellate and similar in size as in Eulothrix, or non-flagellate, non-motile, but similar in size as in Spirogara. Such reproduction is called isogamous. Fusion of two gametes dissimilar in size as in species of Eudrana in term as anisogamous. Fusion between one large non-motile static female gamete and a smaller motile male gamete is termed oogametes, for example, volvox or fucus. This figure is volvox, this Eulothrix. Figure 3. Rhodophyceae, common name green algae, brown algae, red algae. Major pigment chlorophyll A, B, chlorophyll AC, fucoxanthin, chlorophyll AD, phycoerythrin. Stored food starch, mannitol, laminarian, floridian starch. Cell wall, cellulose and agin, cellulose, pectin, and flagella. Number and position of interaction: 2 8 equal apical, 2 unequal lateral. Absent habitat: fresh water, brackish water, salt water. Fresh water rare, brackish water, salt water. Fresh water some, brackish water, salt water most. Gamete sexual reproduction is oogametes and accompanied by complex post-fertilization development. The common members are Polysiphonia porifera, figure 3.1c, Gracilea, and Galidium. Bryophytes. Bryophytes include the various mosses and liverwort that are found commonly growing in moist shaded area in the hills. Figure 3.2. Bryophytes are also called amphibians of the plant kingdom because these plants can live in soil but are dependent on water for sexual reproduction. They usually occur in damp, humid, and shaded localities. They play an important role in plant succession on bare rocks and soil. The plant body of bryophyte is more differentiated than that of algae. It is thallus-like and prostrate or erect and attached to the substratum by unicellular or multicellular rhizoid. They lack true root, stem, or Leaves, they may possess root-like, leaf-like, or stem-like structure. The main plant body of the bryophyte is haploid. It produces gamete, hence is called a gametophyte. The sex organ in bryophyte are multicellular. The male sex organ is called antheridium. They reproduce by flagellate antherozoid. The female sex organ called archegonium is flask-shaped and produces a single egg. The antherozoid are released into water where they come into contact with archegonium and antherozoid fuse with the egg to produce a zygote. Zygote do not undergo reduction division immediately. The, they produce a multicellular body called a sporophyte. The sporophyte is not free living but attached to the photosynthetic gametophyte and derive nourishment from it. Some cell of the sporophyte undergo reduction division meiosis to produce haploid spore. They spore germinate to produce gametophyte. Bryophyte in general are of little economic importance, but some mosses provide food for herbaceous mammal, birds, and other animals. Species of sphagnum, a moss provide peat that have long been used as fuel and as packaging material for transshipment of living material because of their capacity to hold water. Mosses, along with lichen, are the first organism to colonize rocks and hence are of great ecological importance. They decompose rock, making the substrate suitable for the growth of higher plants. Since moss from dense mats on the soil, they reduce the impact of falling rain and prevent soil erosion. The bryophyte are divided into liverwort and moss. Liverworts. The liverwort grow usually in moist, shady habitats such as bank of stream, marshy ground, damp soil, bark of tree, and deep in the woods. The plant body of liverwort is thalloid. Example: Marchantia. The thallus is dorsiventral and closely appressed to the substrate. The leafy member have teeny leaf-like appendages in two rows on the stem-like structure. Asexual reproduction in liverwort take place by fragmentation of thalli or by the formation of a specialized structure called gamma, singular gamma. Gamma are green multicellular asexual bud which develop in a small receptacle called gamma cup located on the thalli. The gamma become detached from the parent body and germinate to form new individual. During sexual reproduction, male and female sex organs are produced either on the same or on different differentiated into a foot, seta, and capsule. After meiosis, spores are produced within the capsule. These spores germinate to form free-living gametophyte moths. The predominant stage of the life cycle of moths is the gametophyte, which consists of two stages. The first stage is the protonema stage, which develops directly from a spore. It is creeping, green, branch, and frequently filamentous stage. The second stage is the leafy stage, which develops from the secondary protonema as a lateral bud. They consist of upright cylinder axis bearing spiral arranged leaf. They are attached to the soil through multicellular and branch rhizoid. This is a stage where the sex organ vegetative reproduction in moss is by fragmentation and budding. In the secondary protonema, in sexual reproduction, the sex organ antheridia and archegonia are produced at the apex of the leafy shoot. After fertilization, the zygote develops into a sporophyte consisting of a foot, seta, and capsule. The sporophyte in moss is more elaborate than that in liverwort. The capsule contains spores. Spores are formed after meiosis. The mosses have an elaborate mechanism of a spore dispersal. Common example of moss are Funaria polytrichum and sphagnum figure 3.2. Pteridophyte. The pteridophyte include horsetail and fern. Pteridophyte are used for medicinal purpose and as soil binder. They are also frequently grow as ornamental evolutionary. They are the first terrestrial plant to process vascular tissue, xylem and phloem. You shall study more about their tissue in chapter 6. The pteridophyte are found in cool, damp, shady places, though some may flourish well in sandy soil condition. You may recall that in bryophyte that the dominant phase in the life cycle is the gametophyte plant body. However, in pteridophyte, the main plant body is a sporophyte which is differentiated into true root, stem, and leaf. Figure 3.3. This organ possesses well differentiated vascular tissue. The leaves in pteridophyte are small microphyll as in Selaginella or large microphyll as in ferns. The sporophyte bear sporangia that are submitted by leaf-like appendages called sporophyll. In some cases, sporophylls may be from distinct compact structure called stabili or cones. Selaginella equistrum, the sporangia produce spore by meiosis in spore mother cell. The spores germinate to give rise to incon species, small but multicellular free living, mostly photosynthetic thalloid gametophyte called prothallus. This gametophyte require cool, dampy, shady place to grow because of the specific restricted requirement and the need for water for fertilization. The spread of living pteridophyte is limited and restricted to narrow geographical region. The gametophyte bear male and female sex organ called antheridia and archegonia respectively. Water is required for transfer of anthrazoid. The male gamete released from the antheridia to the mouth of archegonium. Fusion of male gamete with the egg present in the archegonium result in the formation of zygote. Zygote thereafter produce a multicellular wall differentiated sporophyte, which is the dominant phase of the pteridophyte. In majority of the pteridophyte, all the spore are of similar kind. Such plant are called homosporous. Genera like Reginella and Salvinia, which produce two kinds of spore, macro large and micro small. Spore are known as heterosporous. The megaspore and microspore germinate and give rise to female and male gametophyte respectively. The female gametophyte in this plant are retained on the parent sporophyte for variable period The development of the zygote into young embryo take place within the female gametophyte. The event is a precursor to the seed habit, considered an important step in evolution. The pteridophyte are further classified into four classes: Physlophysia, Physlotium, Leucophysida selaginella, Leucopedium, Sphaenophysida equistum, and Petrophysta, Diopatris patris, and Indium. Gymnosperm. The gymnosperm, gymno-naked sperma seed are plants in which the ovules are not enclosed by any of the wall and remain exposed both before and after fertilization. The seed that develop post-fertilization are not covered are naked. Gymnosperm include medium-sized tree or tall tree and shrub. Example, figure 3.4. One of the gymnosperm, the giant redwood tree squae, is one of the tallest tree species. The roots are generally taproot. Root in some genera have fungal association in the form of mycorrhiza penis, while in some other cycles, small specialized root called colloidal root are associated with N2 fixating cyanobacteria. The stem are unbranched cycles or branch penis citrus. The leaves may be simple or compound in cycles. The peanut leaves persist for a few years. The leaves in gymnosperm are well are well adapted to withstand extreme of temperature, humidity, and wind. In conifer, the needle-like leaves reduce the surface area. Their thick cuticle and sunken stomata also have to reduce water loss. Ploid microspore and megaspore, the two kinds of spore are produced within sporangia that are borne on sporophyll, which are arranged spirally along an axis to form legs or compact strobili or cone. The strobili, being microsporophylls and microsporangia, are called microsporangiate or male strobili. The microspore develop into a male gametophytic germination, which is highly reduced and is confined to only a limited number of cells. This reduced gametophyte is called a pollen grain. The development of pollen grain takes place within the microsporangia. The cone-bearing microsporophyll with ovule or megasporangia are called macrosporangiate or female strobili. The male or female corner strobili may be born on the same tree penis. However, in cycles, male corn and megasporophyll are born on different tree. The megaspore mother cell is differentiated from one of the cell of the nucleus. The nucleus is protected by envelope and the composite structure is called an ovule. The ovule are born on megasporophyll, which may be clustered to form the female cone. The megasporophyll mother cell divided mitotically to form four megaspore. One of the megaspore enclosed within the megasporangium develop into a multicellular female gametophyte that bear two or more archegonia or female sex organ. The multicellular female gametophyte is also retained with megasporangium. Unlike bryophyte and pteridophyte in gymnosperm, the male and the female gametophyte do not have an independent free living existence. They remain within the sporangia retained on the sporophyte. The pollen grain is released from the microsporangium. They are carried in air current and come in contact with the opening of the ovule born in on megasporophyll. The pollen tube carrying the male gamete grow toward archegonia in the ovule and discharge their content near the mouth of the archegonia. Followed fertilization, zygote develop into an embryo and the ovule into seed. These seed are not covered. Figure 3.4 gymnosperm, A cycles, B penis, C ginkago. Angiosperm. Unlike the gymnosperm where the ovule are naked, in the angiosperm or flowering plant, the pollen grain and ovule are developed in a specialized structure called flower. In angiosperm, the seed are enclosed in fruit. The angiosperm are an exceptionally large group of plants occurring in wide range of habitat. They range in size from the smallest volcea to tall tree of eucalyptus over 100 meters. They provide us with food, fodder, fuel, medicine, and several other commercially important products. They are divided into two classes: the dicotyledons and the monocotyledons. Figure 3.5 Angiosperm A. Dicotyledon B. A. Monocotyledon. Summary: Plant kingdom include algae, bryophyte, pteridophyte, gymnosperm, and angiosperm. Algae are chlorophyll-bearing, simple, thalloid, autotrophic, and largely aquatic organisms. Depending on the type of pigment process and the type of stored food, algae are classified into three classes, namely chlorophyceae, phycophyceae, and nodophyceae. Algae usually reproduce vegetatively by fragmentation, asexually by formation of different type of spore, and usually by formation of gamete, which may show isogamy, anisogamy, or oogamy. Bryophytes are plants which can live in soil but are dependent on water for sexual reproduction. Their plant body is more differentiated than that of algae. It is thallus-like and prostrate or erect and attached to the substratum by rhizoid. They possess root-like, leaf-like, and stem-like structure. The bryophytes are divided into liverworts and mosses. The plant body of liverwort is thalloid and dorsivälter, whereas moss have upright slender axis bearing spirally arranged leaf. The main plant body of the bryophyte is gamete producing and is called a gametophyte. It bears the male sex organ called antheridium and female sex organ called archegonium. The male and female gamete fuse to form zygote, which produce a multicellular body called a sporophyte. It produces haploid spore. The spore germinate to form gametophyte. In pteridophyte, the main plant body is sporophytic, which is differentiated into two root, stem, and leaf. This organ possesses well differentiated vascular tissue. The sporophyte bears sporangia, which produce spore. The spore germinate to form gametophyte, which require cool, damp place to grow. The gametophyte bear male and female sex organ called antheridia and archegonium respectively. Water is required for transfer of male gamete to archegonia, where zygote is formed after fertilization. The zygote produces sporophyte. The gymnosperm are the plant in which ovules are not enclosed by any ovary wall. After fertilization, the seed remain exposed, and therefore this plant are called naked seeded plant. The gymnosperm produce microspore and megaspore, which are produced in microsporangia and megasporangia borne on the sporophyll. The sporophyll, microsporophyll, and megasporophyll are arranged spirally on axis to form male and female corn respectively. The pollen grain germinate and pollen tube release the male gamete into the ovule, where it fuses with the egg cell in archegonia. Following fertilization, the zygote develops into embryo and the ovule into seed. The angiosperm are divided into two classes, the dicotyledonate and monocotyledon.